Welcome back to another Homework Biology Lab video, and we're going to be looking at this one, the concept of microscopes. And why do we use microscopes? We use them because we want to observe the universe that we have never known before prior to these devices. Historically, we had no idea about microscopic things living on our body or within our environment. Right? And that's why discovering how to use lenses to magnify images and make microscopes was such a powerful step forward in understanding the world around us. I'm going to take this little guy here, for example. This is called a water flea or Delphinia. And this little organism here lives in water. It's only around about 1 millimeter in size, and it's pretty cool. It sort of swims around. It's got like little legs here that sort of go backwards and forwards and help it move, and you can see a bit up here as well. It's got a compound eye there. Its mouth is about here, so it takes in food, okay, and there's just like this digestive tract down here. It's got a little section at the back there where it can store eggs. So it's pretty cool, but it's extremely small, and you would not know that that was there in water. In fact, if you went swimming in the water, you'd probably swallow some of these guys and increase your protein intake. Right? There's a lot of microscopic organisms in water that we swallow that we don't realize that we swallow. And so this is one of them. But there are so many other ones. For example, this is called a paramecium. It has small little hair-like projections that move backwards and forwards and help it swim through the water. Okay, it's also microscopic. We don't see it. This one here is called a euglena. We've got an amoeba here, which is sort of like a very, it's like a very, how could I say? It's like the blob, if you will. It sort of has pseudopodia, we call it, where it can sort of move, and it's really a cool organism there, right? And then we have bacteria over here, different types of bacteria. We had no idea about these guys. In fact, in history, back in the days of knights and King Arthur and so forth, when they used to cut themselves, they had no idea about germs. They used to think that your body was made up of a variety of things, and one of those things is like normal earth power, right? Because we came from the ground in the sense that we are, you know, living beings just like a tree, and we've got this special life energy. They think that when they cut you, you need to put some of that back. And so they used to put horse manure on cuts and then wonder why, for example, people died. They didn't know about germs, but we do now. So what is this idea about cells and cell size? Well, cells can be large. The largest cells that we have is a nerve cell in our leg, right? But a lot of our cells are microscopic. We can't see them. So what does that mean? Well, microscopic means that it's smaller than you can see with your unaided eye or your naked eye. Now, what does that mean? Well, it means that your eye has no help in helping you see something. Now, of course, I've got glasses on here because you know, my eyes do need a bit of help to make things clearer, but they don't enable me to see things that are microscopic. So an unaided eye or a naked eye is when you're just using your normal vision to try and observe something. If, however, you use lenses that magnify the image larger than what they normally are, that is when you have an aided eye. Okay, and so anything that's microscopic cannot be seen with an unaided eye. So let's have a look here. This is a basic microscope. Don't worry about the names of the parts. We're going to look at that in later videos. But this is a device, right, that you commonly see in school labs that we use to magnify images larger than what they are. And so microscopes are devices that are used to see microscopic organisms or other objects around. For example, the tip of a ballpoint pen, or you might want to investigate soil. If you're a forensic scientist, you want to pinpoint your suspect to a crime scene, so you look at the soil that you scraped off the bottom of their shoe, and you might find that there's certain types of pollen in the soil, or there's certain types of other inorganic matter, bits of sand and stuff that you can say, hey, that's the same sand as the crime scene. So you might be doing that. But the whole idea with a microscope is that you're magnifying things. Now, of course, all these new terms you're writing down in your book, you're pressing pause with the video every now and then and making sure you get the details down to elaborate your points that I'm making here. But remember, in your column, you've got highlights for key words that you don't know. And of course, one of them would be magnification. And that is the ability to produce an image larger than its actual size. And so that's what microscopes do. But there's different types of microscopes, right? So here's a flow diagram that you're probably going to copy into your book, because we want to organize our information in a very visual way. Using color would be good too, and then we can add information to this as we go on. So if I have a look at all the microscopes now, I can group them into two categories. One that uses light to help us see, to help us make things bigger, and then another one uses electrons, and we'll learn about them later. But those electrons enable us to see really close-up views of hair follicles, bits of skin, like so much detail. It's incredible, right? Blow the eyeballs out of your head. So if we look at the microscopes here, these light microscopes can be broken into simple and compound. Well, what does that mean? Well, simple means one lens, right? So write down one lens, whereas compound, you've got more than one lens, right? So two or more, right? So two or more lenses. So to compound something means to add something, right? You might have heard the phrase, you're compounding the problem, you're making the problem worse. When we use compound interest in maths, we're adding interest on top of interest. When we're using a compound in terms of chemistry, we're building it up one atom at a time and making a more complex compound. So to compound something means to build up. And so we're building up our light microscope by using more than one lens to magnify the image. Now, out of those microscopes, we then have monocular, binocular, and stereoscopic, and we're going to look at those ones in future videos. So we have different microscopes for different applications. Okay, so that's it for this video. I hope you've got your notes written down, you've got detail, you've got the flow diagram, you've got your definitions written in your column. You might even highlight those definitions, the particular color you use, because of course, unless you know what monocular, binocular, and stereoscopic is, that should be in your column to go, I need to find out what those words mean. So that's it for this video. I'll see you in the next one, where we continue exploring the science behind microscopy, that is, using microscopes to help us see the world of the unknown universe. See you then.